Okay, this I promise is the final part of Chapter 5, the final video in Chapter 5, because there are only 12 slides to go through. Now, you should have gone through all of your diffusion and osmosis information, and transport proteins are going to heavily involve diffusion, because there are things that are too large to cross a membrane that a cell requires. Things like glucose or even water. They're things that can't cross a cell membrane. Remember, a cell membrane is made of phospholipids. And a phospholipid has a hydrophilic phosphate head, and attached to that head are two fatty acid chains that are hydrophobic. And a phospholipid bilayer is the basis of the membrane. That bilayer is where the phospholipid tails attract each other so that you have heads on either side of the membrane with phospholipid tails making up an interior portion of the membrane. And that hydrophobic interior actually repels polar molecules from being able to cross the membrane. So anything that's polar is going to have a hard time just crossing the membrane. So when it comes to things that are too large to directly cross a membrane, or things that are polar, you're going to need a way to get them across, because a cell needs a lot of those things to live. And the answer to that is the transport proteins. They allow hydrophilic molecules that would be repelled by the hydrophobic interior to pass. They allow polar molecules to pass. And they allow molecules which are too big to directly cross the membrane to pass. There are three types of transport. So, passive transport involves a type of protein called a channel protein. Now, passive transport can also describe the movement of small enough particles that the movement of particles small enough to directly cross the membrane. But when it comes to transport proteins, passive transport describes a kind of transport protein called a channel protein. So that is movement of a substance across a membrane without using energy. And it moves according to the rules of diffusion. Next up is facilitated diffusion. Now, just looking at the name, we know this kind of transport protein is going to move substances along their concentration gradient. It's going to follow the rules of diffusion, just like passive transport. Facilitated diffusion involves the movement of molecules across the membrane via a carrier protein. What is a carrier protein, you might ask? It's a protein that can only bind a specific kind of molecule. And this is what you see most often when you're moving something that's too big to diffuse across a membrane, too big to move through a channel protein. So this is facilitated diffusion. Now, both passive transport and facilitated diffusion move things along their concentration gradient. They follow the rules of diffusion. Because they follow the rules of diffusion, they do not require energy. So they do not require energy. The third type of transport protein is involved in a type of transport called active transport. Now, active transport moves substances against their concentration gradient. It moves them from areas of low concentration to areas of high concentration. This violates the rules of diffusion, and whenever you're going to violate something, you're going to need to invest energy in it. So, passive transport. One of the ways you can have passive transport is diffusion directly across the membrane. This would be very small, non-polar molecules or atoms. Things like two oxygens bound to each other. They're a neutral atom, and so they're going to be able to pass there. Very small particles can cross the membrane directly Channel proteins help slightly larger molecules to pass, and they help polar molecules to pass. So for instance, ions, which are positively or negatively charged, would need to they can't diffuse directly across the hydrophobic interior, so they can use a channel protein to cross. To avoid the hydrophobic interior, the channel protein crosses the entire width of the membrane, and it's just like a little canal that allows things to pass through it. So here's a channel protein. You can see it's like a little tube or a canal, and something can just pass from one side of the cell to the other, according to the rules of diffusion. Channel proteins really only allow the only sort of mechanism they have to prevent things from crossing, to control what crosses the channel protein, is the size of the channel. If it's too large to pass through the channel, then it can't pass through the channel. So these are things that are slightly too big to directly diffuse across the membrane, and things that are polar. So, a little more detail on channel proteins. The canal allows movement past the hydrophobic interior. Water moves through a channel protein called an aquaporin. When you think of a pore, you can think of a little hole or passage. So an aquaporin is a hole or passage through a membrane, and that's in the form of a channel protein. And then ions move through ion channels. So that's incredibly creatively named. So we've got a couple of different channel proteins here. So this is a channel protein that is just always open, and whatever can fit through the channel protein can cross. But here's another kind of channel protein that I find very interesting. This is a gated channel protein. A gated channel protein has a little funky extension, almost like a flap. And this flap can either open or shut. And when it's shut, things can't cross the channel. So it's useful to be able to shut something in case, for instance, you have an ion concentration inside the cell that you need to maintain. You shut that gate, and then ions can't leave the cell. So gated channel proteins exist, and that's just neat. Okay, next up is facilitated diffusion. Facilitated diffusion works according to the concentration gradients of the molecules involved. They move from high concentration to low concentration. Because this is diffusion, it costs no energy, just like passive transport costs no energy because it's diffusion. But in facilitated diffusion, well, facilitate means help. So something is helping diffusion occur. And that something is a carrier protein. And a carrier protein can bind to a particular molecule. It has a binding site. And that binding site is another lock and key thing, where the molecule is the key and the carrier protein is the lock. And if the key fits the lock, it binds to the carrier protein. And when it does that, it actually changes the shape of the carrier protein. And that shape change allows the molecule to move across the membrane, which has an awesome scientific term, translocation. It sounds like a spell Gandalf would use. He'd be like, I translocate you, with his staff. Anyway, that was lame. Leave me alone. So carrier proteins move molecules back and forth across a membrane based on high concentration versus low concentration. So this is extremely useful because you have specificity here. This is going to be moving large molecules. You don't want to necessarily move just any large molecule. So that specificity allows you to move only specific large molecules. For instance, a glucose transporter is an example of a carrier protein. In fact, a glucose transporter is generally inactive in a cell, but in the presence of insulin, when insulin binds to a protein on the outside of a cell, that protein is activating an enzyme which activates the glucose transporter and causes the cell to move glucose into the cell, because insulin is only released when there's a high concentration of glucose outside the cells, which translates into a high concentration of glucose in the blood. High blood sugar. You release insulin. Insulin activates the glucose transporters. The cells use the glucose transporter to bring glucose out of the blood into the cell. That lowers blood sugar. Pretty awesome. So, facilitated diffusion. Again, we're using the rules of diffusion. No energy needed. So let's go to the picture. Here's our, hey, come back here. Here's our molecule binding to the carrier protein right there. You can see it only fits, like this little binding site is specific to that molecule, and the binding of that molecule alters the shape of the protein. You can see now this area is too small for the molecule to exit, so the molecule is able to cross the membrane. That's how it works. When this molecule is released, by the way, that binding site is now open, and an empty binding site causes this protein to revert back to its original shape so that it can bind to another molecule. So, another cyclical thing. Kind of cool. All right. Finally, there's active transport. Active transport moves against the concentration gradient. So we're going from areas of low concentration to areas of high concentration. That's a violation of the natural order of things, so we need energy. So that requires energy, and the energy currency of a cell, what a cell uses whenever it needs energy, is a molecule called... what? That's right! Adenosine triphosphate, or ATP. Active transport requires energy in the form of ATP. So, this is the sodium-potassium pump. It is the gold standard example of active transport. Any biology class you'll take, anywhere in the US, maybe even the world, is when it introduces you to the concept of active transport, going to talk about the sodium-potassium pump. So, this is your active transport protein. And it has these open binding sites that allow sodium to bind. And here they come and they bind there. These binding sites are for potassium, but at the moment, because of the shape of this protein, potassium isn't going to bind very well. So it's really only sodium that can move from the inside of the cell, which is this yellow portion, into the active transport protein. And it binds to the binding sites on my active transport protein. But sodium is in a high concentration inside the cell, is in a high concentration outside the cell and a low concentration inside the cell. So because we're trying to move against the natural order of things, against diffusion, nothing happens when sodium binds. In order to induce the change in shape that will allow sodium to move across the membrane, we have to use ATP. So we hydrolyze a bond in ATP here and remove a phosphate. And in doing so, we release energy. That transport protein is going to use that energy. Now it is the binding of phosphate to this little phosphate binding site. See the mouse cursor? This is a phosphate binding site. When phosphate binds to this binding site, that causes the shape change. And so the shape change occurs, and sodium is no longer very good at binding to these sites, so it's released outside the cell. We've successfully moved sodium against the natural order of things. Now that it's facing outside, these potassium binding sites right here are much better at binding potassium. So in step 4, potassium is going to bind here. So potassium comes along and binds. And once potassium binds, that causes the protein to release phosphate. So the active transport protein releases the phosphate. Once the phosphate is released, well, it was the phosphate that allowed the shape change in the first place, so we revert back to our original shape here. This original shape wasn't very good at binding potassium, so the potassium's released. And it's great at binding sodium, so sodium comes back in and binds. These phosphate and the ADP here, my adenosine diphosphate, are then recycled in order to make some more ATP, and we're ready to go again here. So that is active transport. So what about things that are too big to move using proteins? So molecules that are too large to move across the proteins. Well, you have vesicles. And when you're using vesicles to move something from the outside of the cell to the inside, or from the inside to the outside, you have two very specific terms, endocytosis and exocytosis. Endocytosis is when the cell membrane bends inward, and that bending inward sucks molecules into that little depression, and then that depression goes deeper and deeper until it forms that vesicle. And then the vesicle takes the molecules into the cell where they need to go. Now, if the cell is just trying to kick molecules out, it undergoes exocytosis, which is pretty easy. Exocytosis is the reverse of endocytosis. The vesicle comes along from inside of the cell, and because the vesicle is made, has a phospholipid bilayer, and the cell membrane has a phospholipid bilayer, when the vesicle reaches the cell membrane, here's my vesicle, here's my cell membrane, it's going to fuse to the cell membrane and open up, and that releases the molecules to the outside of the cell. So here we go. This is endocytosis. The membrane bends inward, molecules are sucked into the depression, and then the vesicle forms and is released into the cell to transport those molecules where they need to go. And here's exocytosis, where the vesicle arrives at the membrane, starts to fuse, and then releases the molecules outside of the cell. And here is a very cool microscopic, this is a microscopic series of pictures of an actual vesicle forming. This is endocytosis. So out here are a bunch of little molecules, these dark smudges, and they need to come inside the cell. Well, here's an actual microscopic image of a cell membrane depressing, and it continues to depress inward until it forms that vesicle that pinches free, and now things are loose, or now the vesicle is free to move inside the cell to take those molecules where they need to go. The most likely thing that's going to happen is lysosomes are going to fuse with this vesicle, release their digestive enzymes and acidic fluid, and digest the molecules within there. All right, now we'll take one last look at the second step of active transport so I can deliver a wonderful closing statement for this chapter. That is the end of Chapter 5. Thank you for coming along in this wonderful journey through bioenergetics with me, learning of the magic of osmosis and diffusion and transport proteins. May you all have a good night, study hard, and be smart.